Aging blood stem cells restored to youthful state in mice. Scientists have demonstrated the reversal of aging in blood stem cells in mice, potentially paving the way for treatments that rejuvenate immune systems and combat age-related blood disorders. The research, conducted by teams at the Icahn School of Medicine at Mount Sinai and Paris Cité University, shows that restoring proper function to cellular recycling centers called lysosomes can revitalize aging stem cells, making them behave more like their younger counterparts. The problem with aging blood stem cells, hematopoietic stem cells, HSCs, are responsible for generating new blood cells throughout life. As we age, these cells become less efficient, leading to weakened immunity and increased susceptibility to conditions like anemia and cancer. This decline isn't simply a matter of cells dying off. It's a functional deterioration, where HSCs lose their ability to effectively produce balanced blood cell populations. This matters because a failing blood system directly impacts overall health, making the elderly more vulnerable to infection, disease, and slower recovery times. Lysosomes, the key to reversal. The study pinpointed lysosome cellular compartments that break down waste and recycle materials as the primary driver of HSC aging. In elderly mice, these lysosomes were found to be abnormally acidic and hyperactive, disrupting cellular metabolism and genetic regulation. This hyperactivity prevents the cells from entering a protective quiescence state. Quiescence is crucial because it allows young stem cells to conserve energy, avoid DNA damage, and maintain their regenerative capacity. Restoring youthful function, researchers used a chemical called concanomycin A to normalize the acidity and activity levels of dysfunctional lysosomes in aged HSCs. When treated cells were reintroduced into older mice, blood cell production increased eightfold. The rejuvenated HSCs not only regenerated more efficiently, but also restored balanced blood cell ratios, reversing the age-related decline in immune function. This suggests that aging in blood stem cells isn't a permanent condition, but rather a reversible state of cellular dysfunction. Implications for human health. While the study was conducted on mice, the findings have significant implications for human medicine. Aging HSCs are poor candidates for stem cell transplants, but the treated cells showed successful engraftment in the animal model. This suggests that ex vivo treatments, where cells are modified outside the body before transplantation, could dramatically improve transplant success rates in humans. Our findings reveal that aging in blood stem cells is not an irreversible fate, said lead researcher Saghai Ghaffari. Old blood stem cells have the capacity to revert to a youthful state. The research highlights lysosomal dysfunction as a central mechanism in stem cell aging. Targeting this pathway may eventually lead to therapies that maintain healthy blood and immune systems in the elderly, improve stem cell transplantation outcomes, and potentially reduce the risk of age-associated blood disorders. Further testing is needed to confirm these results in humans, but the study offers a compelling new avenue for combating the debilitating effects of aging on blood and immune function.